Get professional development, there's also extra general classes you need to get your degree. And then she showed me how to pick my classes and make a schedule. Start November 1st. She advised me not to take suicide. Like, an ability that I know, has been having to mental health, because it's not at all. But if you finish all those general education, all those.
Okay, let's go ahead and get started. We are going to continue our discussion on metabolism today and wrap this chapter up, and that will finish our second unit, which will be cells and metabolism. And we won't have a test on this until after everybody gets the bone test done. The bone test isn't due until the end of next week. So today's the what, the 13th?
Yeah.
Okay, so it's not due till the 20th, 21st, somewhere in that range there. I think it's to Friday night next week. So we're going to talk about metabolism, and I'll give you a heads up if you get confused, let me know, because this is an easy topic to get confused on, and I will do my very best to confuse you completely here. I will do my best to get you understand what's going on here. It's the topic area on metabolism. It's something that it sort of underlies this whole area here on how we get the energy from our cells. Sort of underlies everything we do, and it's not the kind of thing, you know, where someone will come up to you and say, well, what do you think about glycolysis in the cytoplasm of the cell? You know, it's not a conversation that comes up, you know, a topic that comes up frequently. But to understand how we get the energy is really important. You know, this is something that has a real impact on what we do, you know, and all the things we do in our bodies. So let's bring this back up now and see if I can open it up correctly. Hopefully everybody can see it now. Okay, now this illustration here, I'm pretty certain this is where we were when we got done the last time. And we were talking, because we take the nutrients, the major nutrients that we consume, the carbohydrates, lipids, and proteins, all have a basis in providing energy for us. They all can be used for energy. Now, the most important of these three are the carbohydrates. Carbohydrates, you know, the number one test question answer, most important molecule in the body for energy, it's always carbohydrates. It's always carbohydrates. That's what we use them for. We use the carbs, primary source of energy, because we take the carbs and break them down to glucose. So whatever we eat, whether we're eating proteins like meat or eggs or beans and rice, remember we can have vegetable proteins that give us the same amino acids in combination, whatever the protein is, whatever the carbohydrate, you know, we eat proteins, we eat carbohydrates, we eat fats. Now, proteins generally give us all the amino acids we need for cellular respiration. Well, not for cellular, for protein synthesis. Protein synthesis, say that in a hurry. We know that takes place in the ribosome. That's how we take we eat a piece of cheese and we break it down to take the amino acids out of it, and then we turn that around and make a new protein. Maybe we're making hemoglobin, or maybe we're making hair color, or eye color, or skin color, whatever the protein is we need. We're making it from the amino acids. The amino acids, most of them don't get destroyed. Most of them are recycled into new proteins. In an extreme situation, we can take the protein and convert it to glucose through the process of gluconeogenesis. We see that happening frequently in your patients that are having an extreme diabetic emergency. Their bodies are starving, and they'll start breaking down fats and proteins to get glucose. We see it in a pregnant female. A woman that's pregnant will usually experience what's known as glucose sparing, where she will send most of her glucose that she's getting from breaking down the carbohydrates out to the placenta so it can go to the baby. And she will continue to get her glucose by breaking down fats and proteins. It gives her a pretty elevated glucose level, and it's sort of the basis of what we call gestational diabetes, where the blood glucose levels or blood sugar is very high, but that's a temporary thing. Anyway, we get proteins and we get amino acids from that. You can see there, we eat proteins and we get amino acids. The carbohydrates are going to give us glucose. We always break down the carbs into glucose. And whether it's a simple sugar like sucrose or fruit or glucose, you know, like in high fructose corn syrup or something like that, we're always going to get down to glucose. Glucose is the molecule that our bodies use for energy. It's not what we use to carry it around, that's ATP, but glucose is the molecule we use for energy. So everything that we eat can ultimately become glucose. So we absorb our carbohydrates in the small intestine. It goes into the blood supply, goes to the liver to be cleaned up. Then it goes up to the heart to be distributed throughout our body. And we carry the glucose in our plasma, and then it goes into the cells if there's enough insulin available. The fats we eat they're going to break down in our small intestine, right along in here in the first 10 inches of the small intestine. They're going to break down glycerol and fatty acids so they can get absorbed. Once they're absorbed into our blood, they reconstitute themselves as triglycerides. They come back together again. They too can be broken down into, converted into glucose in an extreme case through gluconeogenesis. But we're not worried about gluconeogenesis right now, because when you think about it, gluconeogenesis, the formation of new glucose, the creation of new glucose, still ends up going down the middle pathway here on this chart. Glucose initially gets processed in the cytoplasm of the cell. If we have excess glucose, we store it off, we let it get stored as glycogen. If we have a lot of excess glucose, it goes off and gets stored as fat in the adipose tissue. But let's say we just ate nobody's eating anything today. So you just had a Snickers bar, okay? So you can smile because Snickers, do you like Snickers bars?
Yeah.
Good, okay. She just had a Snickers bar and she's smiling now, okay. What's happening with the sugars in the Snickers bar is that they are going into her cells as glucose. And there's a process known as glycolysis, the lysis of glucose taking place in the cytoplasm, and it generates a little energy. Not a lot of energy, but a little energy taking place right there in the cytoplasm. But it's the products of that glycolysis that are going to lead into the third step. Here, if we consider step one is the digestion, step two is the breakdown of the ingredients, you know, whether it's breaking down amino acids, proteins to amino acids and then making new proteins, or breaking down fats to fatty acids and glycerol. When we're breaking down the ingredients, well, that's what we're doing here with glucose. We're breaking down, the chemical reaction, glycolysis. We're breaking apart the glucose molecule to get some energy out of it. Now, this glycolysis step in the cytoplasm isn't very powerful. We don't get a lot of energy from it, but it gives us the products from step 2 become the reactants in step 3. The third stage of cellular respiration is where we get a massive payoff of energy. We get probably 20 times the amount of energy in the third stage that we do in the second stage. So what do you need to remember about all this? Glycolysis takes place in the cytoplasm. That's all you need to remember. Step 2, glycolysis always takes place in the cytoplasm. So I got to remember about this. More to come on that. So, okay. Metabolic reactions in our bodies give us energy. We know what energy is. We talked about it before. I'm not going to beat this into the ground. But the whole purpose of a metabolic reaction is to either release energy or gain energy. If you are if you carbohydrate, if you're a marathoner and you carbohydrate load, you're going to be adding those carbs into your body so you can use that stored energy. You're putting it into your cells. You're putting it as glycogen. That is a metabolic reaction. You're parking that energy there, because metabolic reactions either park energy or use energy. I mean, think about putting gas in your car. In a way, that's a metabolic reaction. It's not, I know it's a stretch, but just indulge me. You're putting gas in your car, you just put energy in there, and you can use that stored energy later on. When you burn the gas, when your engine burns the gas, it releases energy. So, we gain energy, we lose energy. We gain energy, we lose energy. You eat a meal, you gain energy. And as your body goes through the day, you're using the energy up and you lose energy. At the end of the day, you might be tired. And then you eat something, you eat a meal, and you feel better, generally, and we go back and forth like this all the time. So we're using, the metabolic reactions are always going to be about either using energy or acquiring energy. Now, the process of releasing energy, the overall term here is oxidation. Oxidation is when we are going to break oxidation occurs when we break covalent bonds and we release electrons and we release energy. So like even lighting a match here, double match. There we go. Okay, this is oxidation. I have broken the bonds in the chemicals in the head of the match, but now I'm burning the wood. The wood is burning. The wood is undergoing oxidation because it is releasing electrons and it's releasing energy. Every time you break a covalent bond, you're going to probably would be a good idea to put it on paper. So every time you break a covalent bond, you release the energy that's holding the electrons in place, because electrons are being shared in a covalent bond. Every time you break that bond, you release energy. In this case, it's coming off as heat and light. You are breaking the bonds in the wood of the match, or if it was a paper match, it'd be in the bonds in the paper. If it was a lighter, you'd be breaking the bonds in the butane or the propane fuel in the lighter. Doesn't matter. You're releasing energy there. You're releasing it as heat and light. That's a release of chemical energy. We want our body, and it's called oxidation. We want our bodies want to do the same thing. Our cells need to oxidize glucose to break the bonds in the glucose molecule and release that energy so we can use it. But we can't release it all at once. There's a lot of energy in a molecule of glucose, an awful lot. And if we released all the energy and all the glucose molecules in our body at one time, it would be extremely unpleasant for us and for the people around us. Probably explode. So oxidation is the mechanism we use to release the energy. The term that covers all this is called cellular respiration. Cellular respiration allows us to take the energy from glucose and park it temporarily in ATP for 6 or 7 seconds so we can do something with it, so it can power our muscles. Now, obviously, if ATP only lasts for 6 seconds, we got to make it again and again and again, and we do. We're constantly going after it. Okay, so cellular respiration. 3 parts of cellular respiration. We saw it on a diagram here. Digestion, and that's leads into these reactions that take place. We eat food, we digest the food, and then we move in. If it's a carbohydrate, we move into the first part of that called glycolysis. Glycolysis, we start the oxidation of glucose. It takes place in the cytoplasm. Glycolysis is always in the cytoplasm. Can't say that enough. The second stage is what we call the citric acid cycle, or the Krebs cycle. This is where we obtain a little more energy and we release a lot of carbon dioxide. In fact, all the carbon dioxide we breathe out comes from the Krebs cycle. And then the third stage, the electron transport chain, gives us the big payoff. This is where most of our energy comes from. Now, glycolysis has one big advantage. It doesn't require oxygen to work. It's what's known as an anaerobic chemical reaction. Just use glycolysis for that. We find, when we get into muscles, we find that glycolysis can power activity for about 30 seconds. So, when you start doing something, when you start walking down the hall or walking out to your car, if you were sitting downstairs and you started walking, or maybe you were in the library and you got up and walked down out to your car, and 30 seconds is a pretty long time if you actually think about it. That first 30 seconds that you spend doing something, you're running strictly off glycolysis. Electron transport is long-term energy release. You're going to run a marathon. First 30 seconds of your marathon are going to be spent in glycolysis. The last 2.5 hours are going to be in electron transport until you run out of, until your muscle cells run out of oxygen. You know that can happen? There's a thing called oxygen debt. We actually pull all the oxygen out of our muscle cells. We can't undergo electron transport anymore. Electron transport requires oxygen. And then we drop back to glycolysis. Okay, I don't want to get ahead of myself. Now, the end result of cellular respiration is always carbon dioxide. It's always carbon dioxide and water and ATP and heat. 60% of the energy in the food that we eat is lost to us as heat. It gives us a constant body temperature. Our bodies are stuck at 37 degrees C or 98.6. And so the food that we eat, whatever anybody has, or any of the calories in anything that we're eating or drinking, like your juice there, any calories in that? 190. Okay. So 60% of those calories, after you finish that can, or whatever you got at Dairy Queen, oh, well, okay. Well, nothing there. So and that's fine. But 60% of the calories there are going to be lost as heat. But the other 40% are going to be used for something to power your body. So 40%, so we're about 40% efficient, which isn't bad. You know, I think TVA would love to be 40% efficient, you know, on all other things. If our cars were 40% efficient, they'd be wonderful. So, but cellular respiration gives us carbon dioxide and water and energy and heat. So, and cellular respiration has an anaerobic side, doesn't use oxygen. It gives us a little ATP. And it has the anaerobic side, the aerobic side, which requires the use of oxygen. And that gives us a tremendous amount of ATP. So our bodies, our cells can run both without oxygen and with oxygen. They do a lot better with oxygen, but they can run just fine without oxygen. Okay, now the term here, an oxidation reduction reaction. Now this is where we get into chemist talk, and chemists are in a different world. They talk funny. Oxidation is the gain of oxygen. That seems to make sense. I mean, oxidizing something, right? You know, oxidation, we're gaining oxygen. So it means we lose a hydrogen. Now, why are we losing hydrogen? Well, hydrogens have positive charges. A hydrogen atom is positively charged. If it's just a proton, you know, a hydrogen ion is just a proton. It has a positive charge. If we kick off the if we gain oxygen, we generally release the hydrogen. Why do we release the hydrogen? Well, the hydrogen has a positive charge. If we gain oxygen, we kick out an electron. Remember what oxidation is. It's the breaking of the covalent bonds, and we're releasing electrons that release the energy. Well, every time an electron leaves, it takes a hydrogen with it. They travel together because hydrogens are positive and electrons are negative. So every time we oxidize anything, I lit my match, I burned it, I released heat and light, and I released carbon dioxide, and I released energy because we're releasing electrons. Every time we burn something, we're releasing electrons. Now, the chemical reaction, an oxidation reduction reaction, if something is oxidized, it gives up electrons and gives up energy. Can anybody dispute that the match gave up energy and it gave up electrons, you know, when it was burning? Every time we burn something, oxidation, something that's oxidized is going to lose electrons and lose energy. Now, what's a common form of oxidation we might encounter? Anybody ever see anything rusty? Rust is oxidation. The only difference between this and a rusty shovel that you leave out all winter is time. This is a little faster than that. But what happens to the shovel that oxidizes over, that rusts over the winter? You go to use it next spring, and what happens? It might break. Anybody ever have a shovel or a hoe or something like that break on you because it's so rusty? Yeah, there you go. There's nothing as exciting as going to take that first dig with the shovel and the whole bottom of the shovel just sort of folds up on itself. Or you go to use a hoe and the bottom of it snaps off. Whoops, rusty, oxidation, rust, the metal is acquiring oxygen. It's giving up electrons, it's giving up energy, the metal gets weaker. The bonds that are holding the iron implement together become weaker. And so the only difference between a rusty shovel and a burning match is the speed of the oxidation. If we compare this to the combustion of gas in your engine, that's even faster. How about a firecracker? That's even faster than that. But they're all oxidation. They're all releasing energy. They're all releasing electrons. Anything that's oxidized will give up its electrons and give up its energy. We want to take the electrons out of glucose so that we can get the energy out of glucose. Now, because the universe demands balance, whenever we lose electrons and give up energy, something has to gain those electrons. And something has to gain that energy. That energy has to go somewhere. So when we oxidize glucose, when we break the covalent bonds in glucose and we release the electrons, they have to go somewhere. The energy has to go somewhere. Everything has to stay balanced. So we have what we call this oxidation reduction reaction. When something is oxidized, it gives up electrons. Something grabs those electrons. And in the case of the match, it was oxygen. Oxygen grabbed the electrons coming off the match. Oxygen grabbed those electrons. And the energy from that was being released then becomes heat energy and light energy. And we can, if we're holding a match up to see where we're going, well, nobody does that anymore, but in the old movies, you always see somebody lighting a match and saying, oh, it's a gas leak. Whoops, which is nothing more than an oxidation reaction. So oxidation and reduction. Chemists say that things are reduced. Chemists say that things get reduced when they gain electrons. Only a chemist would describe reduction as gaining something. But when something gains more negative charges by gaining more electrons, it says it's reduced. So, okay, we'll just go with that. So now to confuse it even more, whatever is giving up the electrons is the reducing agent. So it's giving up the electrons to have to go somewhere, so the somewhere can become more negatively charged. The acceptor is the oxidizing agent. In our case, oxygen is always the electron acceptor. Just circle that last bullet there and just say oxygen is always the electron acceptor for us. So, and if you weren't confused before, this will really help a lot. Oxidation and reduction. You have your glucose up there in A with its electrons. It's covalent bonds holding the glucose molecule together. It's got electrons. Now we can rip apart those covalent bonds, and we're now oxidized. And we've pulled off the electrons. We've released the energy. We are no longer glucose. We're going to be carbon dioxide and water. Down here, this is oxygen. Oxygen will allow the glucose to give up those electrons. Oxygen will grab those electrons. So we say the oxygen is reduced. It has become more negative. This is why we call it an oxidation reduction reaction. So oxygen grabs the electrons. The energy that we're taking out of glucose ends up attached to the ATP molecule. So this one slide here summarizes redox reactions for you. You're transferring, you're moving the energy from glucose to ATP by using a phosphate. Remember, ATP has 3 phosphates. You are attaching the 3rd phosphate onto an ADP, adenosine diphosphate molecule. And so we're taking the energy from oxidation to force a 3rd phosphate onto ADP. That's where we transfer the energy. When we give up electrons, we're oxidizing. When we gain electrons, we're being reduced. And at the bottom sentence there, adding electrons is called reduction because it reduces the number of positive charges in the accepting molecule. This is an oxidation reduction reaction. We're using oxygen to, we have a candle burning right here. We have several candles burning. This reaction is breaking the covalent bonds in the paraffin, the wax of the candle, and it's releasing the electrons are being released, and we're releasing energy. We've got heat and light coming off those candles, and ultimately we're using oxygen, and we're giving off carbon dioxide and water. And if I have here we go. Demonstration. Show you that it works. I don't work like that. I hold it. This one. Okay, so this is a candle, just to show you that carbon dioxide really is coming off. Okay, now, if I take this beaker and I put it over the candle, what's going to happen? Candle's going to go out. Okay, so we're going to do this and give it a little time. Come on, candle, you want to go out, don't you? Try it this way. Okay, there we go. Now, what do you see forming? Oh, there it goes. There it goes now. What do you see in the side of the beaker? Can we see it? What? You see it? I know. You know why? So
it got oxygen back to it.
Yes. Now let me do this again, and I'm going to put the camera on this, and then you can watch it. Now, what do you see on the side of the beaker? This band right here. What is that? It's condensed water vapor. It's water coming off of the combustion of the candle. Now, if I light the candle again, okay, so there we go. We're busy oxidizing our candle. And now what do we need for oxidation? We need to have oxygen, correct? Okay, because the oxygen, electrons are just flying off of this candle right now. We can't see them, but they are because it's burning. They have to be burning because it has to be doing that because it's burning. Now, if I put the beaker on here, one of the byproducts of oxidation is carbon dioxide and water. If we see water vapor condensing on the side of the beaker, we know that we're having oxidation taking place. And you can see how it's getting cloudy around this band about halfway around. You can see it's clear, higher, but cloudy in the middle. That's water vapor that has condensed on the inside of the beaker. Now, why did the candle go out? Didn't have enough oxygen. And the reason it didn't have enough oxygen is because carbon dioxide formed inside the beaker. Carbon dioxide coming off of the the end result, along with electrons and water vapor. The carbon dioxide is a heavy gas. It's heavier than air. And the carbon dioxide, as it forms, pushes down on the candle and displaces the oxygen. So if I take it back and do this again, there it goes. It comes back up. This is probably still full of the carbon dioxide. The carbon dioxide is a heavy gas. It's heavier than atmospheric oxygen. And every time I do this, every time the CO2 forms inside the beaker, it's trapped there and it suppresses, it pushes the oxygen out of the way, causes the candle to go out. Now, if I take it like this and let it, sometimes you can get it to, you can fill it up with carbon dioxide and you can actually pour it out and put the candle back out while it's still in the air. So this is what's happening all the time. I got wax everywhere too. Okay, this is what's happening in any kind of oxidation and reduction reaction. We're giving off electrons. We're always giving off carbon dioxide. We always need oxygen to make it work. The electrons that are coming off, of the candle, when the candle's oxidizing, they have to go somewhere. When the oxygen grabs the electrons, it also grabs hydrogen. Remember I said hydrogens travel with electrons. Every time an electron comes off, a hydrogen goes with it. Oxygen has two spaces for electrons in its valence shell. It will grab electrons and bring those hydrogens with it. And all of a sudden you've got two hydrogens and an oxygen. What do you have? Two hydrogens and an oxygen. Water, you get water, H2O. That's why we get condensation in a ring around the inside of the beaker. That's the water vapor that has condensed because the electrons traveling with their hydrogens have combined with the oxygen and they form water vapor. So we, without seeing it actually physically, seeing the electrons physically move, we know it's doing it because we get water vapor in here, and we know we get carbon dioxide because the candle goes out. It's not because we used up the oxygen. There's a boatload of oxygen inside this beaker, but we form enough CO2 to suppress the flame, to push the oxygen away from the flame. So now back to stuff less exciting. Okay, so that's what's happening here with this animation. We're seeing that oxygen's being used, carbon dioxide and water are given off. The carbon dioxide is the breaking down, that's the end result of breaking down the paraffin, the wax. Carbon dioxide is being formed and oxygen is being used because the candle's still burning. And we know that water vapor is being formed because we saw it here in our beaker where it condensed in the side. Okay. Now our bodies are picky. We have what's known as a controlled release in here. Every time we release an electron, a hydrogen has to go with it. So you have an electron and a hydrogen for everything. If we released all the hydrogens at once, it would be unpleasant. So we control things. We strip the electrons and the hydrogens away slowly from the glucose. And we do it in a step-by-step process. We have these molecules that are called hydrogen carriers that grab the hydrogens and indirectly grab the electrons too, because the electrons and hydrogens travel together. If you grab a hydrogen, you're getting the electron to come along. So we don't want this to happen. This is what happens if you have if you don't control oxidation. This is real time, by the way, and this is how long it took. This is the Hindenburg in the late 1930s in New Jersey. You've probably all seen it in a history class or somewhere, but the Hindenburg is filled with massive balloons, massive huge silk balloons inside filled with hydrogen, elemental hydrogen, the stuff that's very reactive. They didn't have access to helium, or they didn't want to buy helium, whatever. And so when something ignited the hydrogen, what you're seeing there is a rapid oxidation. You're seeing the rapid formation of water. You're releasing energy and you're producing carbon dioxide and producing a lot of water all at once. We don't want that to happen in us. So we use molecules to carry the electrons and to carry the hydrogens. So we can make it stepwise. So what are the two important electron acceptors that we use here? They're called NAD plus and FAD. Okay. I seriously doubt if anybody's ever going to ask you again after this semester about NAD plus or FAD, but you might want to make a note of it at least for now. So, cellular respiration. We want to take hydrogens from glucose and give them to oxygen. It's the best way to transfer the energy from a large molecule to a small molecule. Every time we break a covalent bond in glucose, we're going to release a hydrogen. When we release the hydrogen, that releases the electron, because hydrogens and electrons travel together. I don't know why they do that, but they just do. So every time we break a bond, every time I lit my match, every time I burnt the candle, I am releasing hydrogens that are dragging electrons with them because they travel in pairs. And the reason they travel in pairs is because they're balanced. The positive hydrogen is balanced by the negative electron. And it's always the valence electrons that are at work. The valence electrons have the greatest amount of potential energy. So, now here's the formula for cellular respiration. C6H12O6, which is glucose, plus 6 oxygens yields 6 carbon dioxides and 6 waters and a release of energy. This is the basic formula here. Glucose gets oxidized to carbon dioxide. It's going to give up 12 hydrogens. The oxygen is reduced to water. Remember, with reduction, something gains electrons. So oxygen gains 2 electrons and 2 hydrogens, becomes a water molecule. So every time you have cellular respiration taking place, a molecule of glucose will give you, in the presence of oxygen, will give you carbon dioxide and water and a release of energy. So. Now, this is one of those hydrogen carriers, hydrogen acceptors, the NAD. It will pick up hydrogens and electrons with it. So, another important point, protons are hydrogens. Hydrogens always travel with an electron. So the transfer of energy from our food, we take the high energy electrons from the valence shell and bring along their hydrogens. And we do the greatest amount of energy transfer in the electron transport chain. Let's get into glycols. Let's talk about this. So ATP, each ATP molecule, here's a little factoid that is probably useless, but you'll need to remember it for a while anyway. Each ATP molecule contains 7.3 kilocalories of energy. Now, there's two ways to park energy from food into ATP. These you need to remember, at least for now. Substrate level phosphorylation. This is what takes place in glycolysis. Substrates are what we use in are what the reactants are with an enzyme. And so we're using enzymes to phosphorylate something. We're transferring the phosphates. Phosphates are how we're packing the energy into the phosphate and attaching it on to ATP. So substrate level phosphorylation. Oxidative phosphorylation is the big payoff. That takes place in electron transport. So substrate level in glycolysis, oxidative phosphorylation in electron transport. Make sure you know those two, at least for this semester. After that, it doesn't matter. So this is substrate level phosphorylation. Glycolysis and the Krebs cycle do this. This is where we have an enzyme taking an enzyme will have a substrate with a high energy phosphate and attach it to ADP. Now we've got ATP formed. Substrate level phosphorylation. It's not very efficient. Doesn't give you a big payoff. But this is what we do in glycolysis. And the first step of cellular respiration, glycolysis in the cytoplasm uses substrate level phosphorylation. In the mitochondria, though, we have oxidative phosphorylation. The electron transport chain moves electrons around so that we can use their energy, and we transfer those electrons ultimately to oxygen. And the energy comes off to produce lots of ATP. So this is how instead of having enzymes just with the substrates, this is how we generate ATP inside the mitochondria. Embedded in the membrane of the mitochondria, there's a structure here called the ATP synthase. It's an enzyme. And what we're doing is as we bring, this sounds really, really bizarre, but we have lots of hydrogens out here in this intermembrane space. We've pumped them out there. And as they come back through, they cause this thing to go around in a circle, and it physically forces the third phosphate with its high energy onto an ADP molecule, and we make a lot of ATP this way. Every time I talk about it, I'm amazed that someone was able to figure out how this works. Because we're talking something that's inside the membrane of the mitochondria, which is an organelle to begin with. So it's extremely tiny, and this is even smaller. So let's try not to bore you too much in this, but let's talk about the 3 steps to get down to electron transport. Glycolysis is the first one. Glycolysis takes place in the cytoplasm, outside the mitochondria, outside the nucleus. Glucose has come into the cell through various channel proteins or carrier proteins, thanks to the role of insulin. Glucose comes in and it gets broken down inside the cytoplasm. So here, important things about glycolysis. It takes place in the cytoplasm. It is substrate level phosphorylation. So it uses enzymes to move the phosphate around. It's anaerobic. It doesn't require oxygen. And the end result of glucose is 2 pyruvic acid molecules. Glycolysis is what a marathoner uses at the end of a race. We store oxygen in our muscles in myoglobin. Myoglobin is similar to hemoglobin. It's what makes our muscles red. And we park lots of oxygen in the myoglobin. We can deplete that oxygen, and we're in what we call oxygen debt. And if you've depleted all the oxygen, it takes about a day for all your muscle cells to get back to where they were before. However, there's still lots of glycogen available. A marathoner who has depleted all their oxygen can still function, can still run off of glycolysis, because glycolysis doesn't require oxygen. So you're at the 25th mile and you have 1 mile and 2/10 to go, and your legs hurt and your arms hurt and everything hurts, but you can see ultimately you can see the finish line ahead. You're running now off of glycolysis. You're burning up the glycogen and releasing a little energy, enough to keep your muscle cells functioning, but you're not releasing a lot of energy because you don't have any oxygen left in your muscle cells to, at least in the myoglobin, to do anything with. So glycolysis doesn't require oxygen. We don't use oxygen when we start some activity. And if we deplete all the stored oxygen, we don't use it at the end of the activity. So here's the end result of glycolysis. It produces 2 pyruvic acid molecules. It gives us 2 NADH molecules. Okay. The NAD has been reduced. It has gained 2 hydrogens and 2 electrons. It makes 4 ATP. Okay, important point. Glycolysis makes 4 ATP. Remember that point. Glycolysis makes 4 ATP. Circle that. Make a star there. Now the next bullet down, it says there's a net gain of 2. Because you make 4, but you had to burn 2 to start the process. Sort of like when you start your car. Your battery gives you that initial spark for your car to turn over. You don't run off your battery after that. If you've ever had a bad alternator, you know what I'm talking about. Yes, thank you, Emma. Alternators are what provide you the electrical energy the rest of the time, but the initial spark to turn over your engine comes from the battery. You spend energy to gain energy. So this is what happens in glycolysis. We have to burn 2 ATP to start the process. So we spend 2 ATP to start glycolysis, but we make 4. So we have a net gain of 2. Okay, so glycolysis gives us a net of what? 14 kilocalories. Each ATP is like 7.3, so 14.6 kilocalories. Yay, glycolysis has given us 14.6 kilocalories of energy. But more importantly, and remember, the ATPs came from substrate level phosphorylation. They were formed simply by jamming a high energy phosphate onto ADP. But the most important thing that happened here was those NADs captured 2 high energy electrons. Each one grabbed 2 high energy electrons. That's much more important to us than just producing a couple of ATPs. Oh, the ATPs are fine. They'll keep us going when we've depleted all the oxygen in our muscles. They'll keep us going indefinitely. And our cells aren't going to suffer because we're still breathing and still transferring oxygen through our blood, but we won't have any stored oxygen for about 24 hours. This oxygen debt takes a while to recover from. But what's more important is we grabbed a couple of high energy electrons in each of those NADs. We call them NADHs now because they've got 2 hydrogens, and every time you have a hydrogen, you got an electron. So what happens next? Well, we're going to take advantage of oxygen now. So I've said this a couple dozen times here. ATP is formed by phosphorylation of adenosine diphosphate. Now, whether we do it with an enzyme, as in substrate level, or we do it oxidatively like we do it in electron transport, it doesn't matter. We're still putting a phosphate onto ADP. So, but 4 ATP or 2 net ATP, that's not very much. We want more. There's your ATP molecule. It is that third phosphate going on there is where we can park energy for about 6 seconds. 7.3 kilocalories right there for about 6 seconds. We send that around, so it moves around in the body wherever we need it. You know, muscle cells produce lots of ATP, and each one gives us about 7.3 kilocalories of energy to do something. Now, but the mitochondria, as most biology textbooks would say, is the power plant of the cell. This is where most of the energy transfer takes place. The energy that's still contained in, we took glucose and broke it down into 2 pyruvic acids. We took a 6-carbon compound, glucose, and broke it into 2 3-carbon compounds. Pyruvic acid. Every time glycolysis takes place, we end up with 2 pyruvic acid molecules. Pyruvic acid has to go into the mitochondria to start up, to complete the process of cellular respiration. Pyruvic acid goes into the mitochondria. It goes into the matrix. The matrix is the middle portion of the mitochondria. Remember, it looks like a little sausage, just like the inside of the sausage. So it comes into the matrix, and the pyruvic acid is immediately oxidized and broken down into something known as acetyl-CoA. At the same time, the 3rd carbon goes off as carbon dioxide. So let me show you here. This is what's happening. Here comes the pyruvic molecule over on the left-hand side. Pyruvate's coming into the mitochondria, it goes into the matrix. As soon as it comes into the matrix, this pyruvic molecule is oxidized. The first thing that happens is we see a carbon being stripped off. There's our first carbon dioxide right there in the mitochondria. These are the only places that carbon dioxide comes from. The mitochondria inside our cells. We also we strip off the carbon dioxide and we pull off some more electrons and some more hydrogens with our old friend NAD, and then we form something called acetyl-CoA. Don't ask me how it happens, because I have forgotten. I'll be the first one. At one time I can tell you step by step by step, because I was really boring in those days, worse than I am now. But just take my word for it. We form acetyl-CoA. I don't think anybody's ever going to ask, hey, tell me about acetyl-CoA. So, but what happens? We pulled off, we've made a CO2. Every pyruvic molecule that goes in, there's 2 of them. Each one will give off a CO2. The CO2 we're breathing out the only place it comes from in our bodies is inside the mitochondria. But we also strip off some hydrogens and electrons up there in that NAD. It's carrying those extra electrons. That's more important than anything else happening right now. So the acetyl-CoA goes into the Krebs cycle. It's a 2-carbon compound, and it combines with a 4-carbon molecule of what we call oxaloacetic acid, and gives you citric acid. So sometimes we call this the citric acid cycle. So here's what happens. There's your pyruvate coming in. It's getting oxidized. Off goes the CO2. We grabbed a couple of high energy electrons. We form the acetyl-CoA, and we go into the Krebs cycle. And I need to go, because this is going to get really tedious. This is the Krebs cycle right here. Now, what do you need to remember with the Krebs cycle? The Krebs cycle generates some ATP. It generates the remainder of our carbon dioxide. Remember the formula for cellular respiration said there were 6 CO2s. Each pyruvate gives you a carbon dioxide. That's 2 there. And then we give off a CO2 here and here. So we get, each pyruvate gives you 1, 2, 3. And the second pyruvate gives you 1, 2, and 3. So you get 6 CO2s for every molecule of glucose. Let's show you the end stage. Here's what you need to remember. The bottom line for the Krebs cycle. The Krebs cycle will give you 6 carbon dioxides. It will give you 2 ATPs. It will give you a grand total of 8 NADHs carrying high energy electrons and 2 FADHs carrying high energy electrons. There's a reason that we're stripping off these high energy electrons. Remember, these are valence electrons. They're the electrons that are in the valence shell of the molecule. We want that energy. So again, we're only getting 2 ATP out of our Krebs cycle. Not a big payoff yet. So what, we got 2 net ATP from glycolysis. We get 2 more ATP here. We're up to about 29 kilocalories of energy, and there's about 680 kilocalories available in a molecule of glucose. So then we go into electron transport. Embedded inside the inner membrane of the mitochondria are a series of enzymes. These enzymes are designed to grab electrons and transfer them from one enzyme to the next enzyme to the next enzyme. The energy of the electrons will pump hydrogens from the middle of the mitochondria into the what we call the inner membrane space. The whole purpose of electron transport is to pump these hydrogens into one location. So this is the electron transport chain. There are 3 enzymes embedded in the matrix. Here's the matrix of the mitochondria. This is the inner membrane here. And these are these enzymes in the inner membrane. Up here is the inner membrane space. Every time an NADH comes up here, it will give up its electrons, and the electrons will be pumped up into the inner membrane space. Better animation than this. Okay. So there's a better picture of the electron transport chain. You see the membrane there. We can see the phospholipids. That's the inner membrane of the mitochondria. NADH will come up to that membrane, and in the membrane we have enzymes. And every time, that's it. Every time an NADH comes along here, the NADH will get oxidized. The electrons we grab, and watch, forget this for a second. That's slow. Stuff. There we go. Okay. On the far left-hand side, here comes the NADH. It's carrying 2 electrons and 2 hydrogens. The hydrogens are pumped across the membrane. The electrons are powering that. The electrons are grabbed by the other enzymes and transferred from pump to pump to pump. And it keeps pumping hydrogens into the inner membrane space. See all those little orange, yellow circles up there? Those are hydrogen ions. Now, these ions want to reach equilibrium with the other side of the membrane. They want to diffuse back through. The only way they can come back through is through the ATP synthase there in green. I don't know if it's green, and when they do that, they spin around and they shove that third phosphate onto ADP. So now, this is electron transport and oxidative phosphorylation. You need to remember that term. Oxidative phosphorylation takes place in the electron transport chain because the hydrogen ions are going to come back through the ATP synthase. They're going to cause that thing to spin around like a pinwheel, and that's going to jam those phosphates onto the ADP. Now, what about oxygen in here? Remember, this is an aerobic process. The oxygen that's present in the mitochondria will grab those hydrogens down in the matrix, and that will form water. The oxygen molecule, the oxygen atom, will grab electrons. It wants to be stable, and it grabs electrons, but those electrons are attached to hydrogens. Now watch this animation. Here come all these hydrogens going through here. We don't want to have all these hydrogens here, so we got to get rid of them two ways. We got to continue to pump them back up into the intermembrane space, or we need to combine them with oxygen. And if there's oxygen available, then these hydrogens that are floating free here, they come through the ATP synthase. We got to get rid of them. Here comes oxygen, grabbing them and making water. Because if we don't get rid of the hydrogens, we reach equilibrium and the chain stops. We don't make any more energy. That's what happens when we die. When we no longer have oxygen available for our cells, the cells stop making ATP. As long as there's oxygen available to grab those hydrogen ions that are coming through the ATP synthase, we can make all the ATP we want. But when we run out of oxygen, then we're in trouble. Now, oxygen is referred to as the final electron acceptor. Make a note of that. The final electron acceptor is always oxygen. And as long as we have oxygen available, then the hydrogen ions, the red and white things there, are going to be making ATP. And how much ATP do we get out of this? About 36 ATP. So we got four total, 2 net in glycolysis. We got 2 from the Krebs cycle, and we get 36 here. So somewhere, different textbooks say different things. Somewhere in the 30s is what we're going to get. It changes every year. So somewhere, 36 is a nice round number. So, okay. And so 3 terms to remember. Glycogenesis, the creation of glucose, the creation of glycogen from glucose. When we have more glucose than we need, we're going to store glycogen in the liver and in our skeletal muscle. That's what's going to continue to give us energy. We have so much energy stored in glycogen that even after death, skeletal muscle cells will stay alive for up to 12 hours after death. The brain's dead, the heart has stopped, but skeletal muscle cells, individual cells, are still alive. It takes them a long time to die. When we get into muscles, we'll talk about why rigor mortis, the stiffening of the body after death, occurs, and why it's a gradual process. It has to do with the final breakdown of glycogen. So, glycogenolysis. When our blood sugar levels are low, we can break down glycogen into glucose. Glycogenolysis is different from glycolysis. Glycolysis is breaking down glucose. Glycogenolysis is breaking down glycogen. And of course, my favorite term, the gluconeogenesis, where we convert the lipids and the proteins into glucose. We can do this when our bodies are depleted of glucose. We depleted of carbohydrates. We can go after the lipids. We see this occasionally in an individual that's having a diabetic emergency, where they're not able to process to get enough glucose into their cells. Their cells are starving for energy, and the cells are not, the cells can't get the glucose in because the insulin's not working, your patient will start converting their fats into glucose because their body believes, well, the reason I'm not getting fed is because I'm not getting any glucose, even though it's, so you get this patient with a blood sugar level of 600, and anything above 100 is of concern. So you have this patient with a blood sugar level of 600 because they can't get it into the cells because the cells aren't accepting it because their insulin's either not there or not working. So the body's response is, hey, well, obviously I don't have enough sugar. I'll just start breaking down my fats. And gluconeogenesis is taking place to convert fats into sugars. And it produces a condition known as ketoacidosis, which is an even more dangerous condition than just the elevated sugars. So we'll talk all about that in AP2. Okay. By the way, this was the worst material, the hardest material that we will cover in AP1. So we're done with it now. So yes, and I promise you we won't talk about it ever again. So.
Good, because you already lost me.
I'm sure I did.
I'm gonna have to go back to that video.
Yeah, well, I know this is not an easy topic. And you should have seen how I used to teach. I got really.